Actually, this time I wanted to, previously I did it from my cabin itself. So now our library has created one fantastic facility, digital library.
Oh,
So I thought all these webinars nowadays I'm doing from here. So without any interruption, sound free, and very nice facilities here.
Umesh got here?
ಸರ್ ನಾನು ಆಂಟನಿ ಇಲ್ಲಿದ್ದೀನಿ ಸರ್.
ಗೊತ್ತಾಯ್ತು. ಉಮೇಶ್
ಎಸ್ ಸರ್.
ಉಮೇಶ್ ಅಂಡ್ ಸಿದ್ದೇಗೌಡ ಸ್ಟಾರ್ಟ್ ಮಾಡೋಣ.
Yes sir, we can start sir. Before that, Anthony, please record it and start live streaming.
Yeah, live streaming is going on.
Yeah, please start recording.
I'll start the recording.
Recording in cloud, okay?
Umesh.
Yes sir.
Use Dr. Arun sir to...
Yes sir, yes sir. Very good evening sir. Umesh here sir. Namaste.
Namaste sir, good evening.
Nice meeting you sir. ನಾನು NITK alumni sir.
ಹೌದಾ? Oh, very so nice. ಯಾವ ಬ್ಯಾಚ್?
Department of Applied Mechanics sir, 2008.
2008
batch sir.
ಹೌದಾ? Okay. ಯಾವುದು B.Tech M.Tech ಮಾಡಿದ್ರಿ?
M.Tech sir.
Okay, nice.
Under the guidance of Dr. Lakshman Nandgiri sir and Abba Shetty madam.
Oh, about water resources.
Yes sir, yes sir.
Nice.
Shall we start sir? Sir, Ramesh sir?
Please start.
Slides, can we share it?
Yes sir, you can. It's given, option is given to you.
No, that sir, that
No.
option is not activated.
It is not activated, Anthony. Yeah, now it is.
Activated.
Now it is activated. Yeah. Yes, sir. I think slides is
Yes sir, it is visible sir.
Visible now, okay.
Please introduce Dr. Arun to...
Yes sir. Very good evening to one and all. First and foremost, I would like to welcome today's speaker, Dr. Arun Isloor sir, for today's lecture on organic molecules in the eve of on the eve of this National Talent Search webinar series. Also, I would like to welcome our CEO, Dr. A.M. Ramesh sir, and my colleagues, Siddhigoda and Anthony, and all the participants. Before beginning the session, I would like to give a brief introduction about Dr. Arun Isloor sir. Dr. Arun Isloor sir has completed his MSc in Applied Chemistry from Bangalore University, and after that he has finished his PhD from Medicinal Chemistry in Bangalore University. Post that he has completed postdoctorate from Technion Israel Institute of Technology in the year of 2005 to 2007. So he has vast experience in teaching and research around 17 years, and his major research areas of interest are membrane technology, medicinal chemistry, organic electronics, nanomaterials, X-ray, crystallography, organic synthesis, etc. He has published many research publications of reputed journals and books, around 265 research publications, and he has attended many national and international conferences, around 70 plus, and his H index of the journal papers are around 44, and I-10 index is around 108 numbers. Present citations are 6038, and sir has awarded 3 US patents and 1 Indian patent. Also, now currently he has filed 5 patents down the line. And sir has published 3 books mainly in green sustainable process for chemical and environmental engineering and science, which is a supercritical carbon dioxide as green solvent in from Elsevier publications. And the second book is Advanced Nanomaterials for Membrane Synthesis and its Applications. And the third one is Inorganic Nanomedicines in the year of 2000, which is published in Switzerland from Transtech Publishers. Sir has guided around 17 PhD students, and currently 7 more students are working under him. And sir is visiting scientist in Technion Israel Institute of Technology, School of Physics, University Science, Malaysia, and University Technology of Malaysia, and University of Waterloo and Canada, and University of Toronto, Canada. Also, sir is the identified as a top 2% of the scientists at the world level, as surveyed by Stanford University in the year of October 2021, and he has been awarded Best Teacher Award in the second position in science by Akademistan, which is a non-profit organization. Among 10,000 plus nominations filed, among that, sir has awarded Best Teacher Award in the second position. And sir awarded by Sir C.V. Raman Young Scientist State Award in Chemical Science on August 6, 2018. And also, sir has been selected as the top 5 researchers in the field of materials research as per the Scopus survey held during March 2014. And sir has received Center of Excellence Award from our own department, sister concern department, VGST, in the year of 2014 for an amount of 60 lakhs. And sir received Certificate of Excellence for reviewing from Desalination Journal of Elsewhere for the year of 2012, and sir has received Best Research Paper Award for the year 2011 from Vision Group of Science and Technology, Karnataka Government, in the year of 2012. Also, sir is a young scientist, received a Young Scientist Award from Board for Research in Nuclear Scientist from the Department of Atomic Energy, Government of India, in the year of 2009. With this brief introduction, I welcome you once again to deliver today's session on organic molecules, sir. Over to you, sir.
Thank you, Umesh ji. I hope I'm audible to all of you.
Yes, sir. Yes, sir.
Okay. Thank you. And let me congratulate the KSTA for organizing such a wonderful program, that is Talent Search, as well as many other programs to motivate our budding scientists through these type of programs. Even I know this is even in villages, every places it's very popular, this type of activities. So congratulations and thanks for motivating our budding scientists towards science subject. Yeah, so for the next, so our timing is now it is 4:46. So around 5:25 or something like that, I will continue, and then 5:25 to 5:30, and after that is about 45 minutes, and then we can some 10-15 minutes for the question and answers. So the topic which I have opted is organic molecules. So dear students, you know that organic chemistry, which is a branch of chemistry and one of the highly rewarded discipline, I can say, in the chemistry itself. It may be in our day-to-day life, maybe any drug molecules, or it may be the oils and fats, or any compound, maybe the plastics or this type of plastic bottles. So all these are because of the organic chemistry. Not only that, even the life system. So through whether it may be the nucleic acids, or it may be the proteins or carbohydrates, amino acids, etc. So all these are made up of organic molecules only. So I have opted this particular topic, that is organic molecules. So here I will be highlighting about what is organic chemistry, what are some simple organic molecules, and then in what way they are influencing in our day-to-day life. So these are some of the contents. And at any moment, if you have any questions or any clarification, immediately you can interrupt and you can get it clear. Okay. So first of all, let me thank Dr. Ramesh ji, as well as Professor Ayyappan ji, and the entire team of KSTA for organizing various very popular events throughout our state. And my thanks to Director of NITK Suratkal for wholehearted support and the encouraging for all these type of activities through NITK. And thanks to Department of Atomic Energy Board for Research in Nuclear Sciences, DRDO, Department of Information Technology, Vision Group on Science and Technology, Government of Karnataka, as well as Ministry of Mines, Government of India for generous funding. So because of their generous funding, we could did some research, and in terms of whether it may be, it has given few patents as well as few publications. And of course, thanks to all my group members, about 17 students who have obtained their PhD from our group. So thanks to all those my former group members for their contribution in the field of organic chemistry as well as polymer chemistry. So I'll be talking about the carbon and the molecular diversity of the life. As you know, the carbon, so it's the atomic number, that is as well as the atomic mass, it is 12, and the living organisms, any living organism, including the microorganisms also, they consist mostly of carbon based compounds. And carbon is unparalleled in its ability to form the large complex as well as diverse molecules. Like the proteins or deoxyribonucleic acid, or commonly termed as the DNA, or the carbohydrates and other molecules that distinguish the living matter, are all these are composed of the carbon compounds only. So carbon it's a very important from even from the life system of this universe. So the carbon is important enough to have its own branch of chemistry called as organic chemistry. So all of you are aware about this one. So organic chemistry, it deals with it's nothing but the chemistry of the carbon compounds. So it's such a vast field. It's such a vast field. Everywhere we can find the presence of the carbon atoms. And the organic compounds, which range from the simple molecules to the very highly complex also. And most organic compounds, they contain the hydrogen atoms in addition to the carbon atoms, with oxygen, nitrogen and phosphorus among others thrown in from time to time. So there are of course the scientific community always needs some experimental data or experimental proofs, and across the globe, many of the scientists, they worked in this field, in this particular domain, to prove that carbon molecule or the carbon atom was mainly responsible for the emerging of life system on this universe. So among that, one such a very popular experiment, that is by scientist Miller and Urey. So they have conducted an experiment. They have demonstrated the abiotic synthesis of the organic compounds. Here what they did was, it was a very simple experiment. The water molecule, or straight away the water, then the methane gas, that is CH4, then the ammonia, that is NH3, and hydrogen, that is H2. These were all in a calculated proportion. These were sealed inside a sterile array, or a sterile glassware, which is fitted with a flask on a loop. And with one flask, half which is filled with the liquid water, and in another flask containing a pair of the electrodes. You'll see that in the next slide. Here you can see, in one side there is you can see the a round flask with filled with the water, and on the other side there are electrodes where the gases, such as on the right side of this picture, you can find the introduction of the methane gas, ammonia gas, as well as the hydrogen. And also it is provided with the condensing system. So the liquid water, which was on the left side of the flask, it was allowed to heated. So water was heated to induce the evaporation, and at the same time simultaneously sparks were produced with the help of the electrodes to stimulate the lighting through the atmosphere and water vapor, and then the atmosphere was cooled again so that the water could condense and trickle back into the first class in a continuous cycle. So this was the experiment which was conducted, and this is one evidence also. You will see in the next slide, you will see that within a day, so this process was continued for a day, more than a day. So within first day itself, the mixture had turned a pink in color, and at the end of 2 weeks, that means it was continued for more than 2 weeks. So by the completion of the second week, the continuous operation, the Miller and Urey, they observed that as much as about 10 to 15% of the carbon within the system was now in the form of the organic compounds. About 10 to 15% the under these conditions, that is heating condition, and on the other side there is a the sparking which is produced through the electrodes, and on the other side, on the right side itself, there is a condensing system. So this system was continued continuously for about 2 weeks. So by the end of second week, about 10 to 15% of the carbon has converted in the form of the different type of organic compounds. And later on analyzing, they found that 2% of the carbon had formed amino acids that are used to make the proteins. Yes, students, as you know, the backbone of proteins, that is nothing but the amino acids itself. So 2% of the carbon was utilized for the formation of the amino acids that are used to make the proteins in the living cells. And among those amino acids which are formed, glycine as the abundantly formed, which is the first member among the these amino acids. Of course, there was nucleic acids formation was not observed within this particular reaction, but the common 20 amino acids were formed in various concentrations. So this experiment, it indicates that this is a purely an artificial conditions which was maintained. Probably the life system was originated a few millions year back on this particular universe, something by this type of natural process itself. So this is just an experimental evidence which is suggesting through the carbon atom itself, all other complicated molecules like carbohydrates, proteins, amino acids, which have been formed. That is nothing but these are all life supporting molecules. So with the 4 valency electrons, as you know, the valency of the carbon is, let's say, 4. It can form 4 covalent bond with a variety of the other atoms. And this particular ability, that means to make a covalent bond with the other 4 atoms. So this makes large complex molecules, different type of the possible complex molecules. And in molecules with multiple carbons, each carbon bonded to the 4 other atoms, which has a tetrahedral shape, as shown in the figure. However, when 2 carbon atoms are joined by a double bond, that is carbon-carbon double bond, the atoms which are joined to the carbons are in the same plane as that of the carbons. Now the question is, why is this important? Because the shape of the molecule, that is actually going to dictate its reactivity, thus its function. So whatever the molecule it may be, whatever the any molecule of the made up of the carbon atoms, so what type of the shape, what is the shape of that particular molecule, that is going to dictate, or that is going to decide the reactivity of the particular molecule, as well as its function also will be decided by that particular shape or the geometry. So we are not going any more details about the those geometry of any of these molecules. Just for your kind information, I have included a few of the organic molecules, simple molecules such as the methane, ethane, ethene. Ethene is it's a C2H4 or CH2 double bond CH2, which is in the molecule C. Okay, and at the same time on the right side there is a structural formula as well as the ball and stick model that is also been given along with the space filling models for these simple organic molecules. Of course, the carbon skeleton, with the increase in the number of the carbon atoms, the chain length of this carbon also, this also go on increases or it enhances. So you see here in the first one there is a ethane C2H6, then the next molecule is the propane. So in the first ethane there are 2 carbon atoms, whereas in the propane there are 3 carbon atoms, that means slightly larger. Then the next one is butane. Of course, below that it has been given. Its unsaturated form is, that is the butane, and based on the position of the double bond, whether it's the first position or second position, these are either 1 butene or 2 butene. So it purely depends upon the number of the carbon atoms and also whether there is any double bond or any unsaturation is present or it's with the saturated type of the bonding. So both these saturated as well as the unsaturated systems which I have presented here. And as you know, the hydrocarbons, these are organic molecules consisting of only carbon and hydrogen. So the whatever the previously in the previous slide, the methane, ethane, propane, butane, or other molecules which I have presented, they are purely the hydrocarbons. That means they are made up of carbon and hydrogen only. And also you can see there was a cyclic compound also, that is the benzene. It is made up of carbon and the hydrogen, and it is in a cyclic structure. You can see there the hexagonal structure. Suppose in this particular structure, in this particular molecule, if there is presence of other than carbon and hydrogen, other atom like sulfur or nitrogen or oxygen or phosphorus, then they are nothing but the heterocyclic compounds. Other than carbon, hydrogen, any other atom is present along with that, like sulfur, nitrogen, oxygen, phosphorus, etc. So they are termed as, and if it is a cyclic compound, then they are termed as a heterocyclic compounds. So these are purely the hydrocarbons. So many organic molecules such as it may be the fats. Basically the fats are triglycerides. They are the esters of glycerides only. They have hydrocarbons as a components, and hydrocarbons can undergo the reactions which release a large amount of the energy. So this is just a fat molecule where the fat droplets, basically the fat droplets, these are nothing but, these are the glycerides, triglycerides, or the esters of the fatty acids. So which is termed as the fat, or oil and fat are nothing but the esters of the fatty acids. Similarly, there is a nucleus and also the other part, that is a part of a human adipose cell. So this is actually the part of the human adipose cell, as well as on the part B, which is the focus one, which is nothing but a fat molecule. So this term, that is isomers, I think since when you are in 8th or 9th standard, this particular terminology you have probably already came across with this terminology, that is isomers. So these isomers are nothing but, these are the compounds with the having same molecular formula, but structurally they are different. Molecular formula is same, but the structure wise they have the different structure. So because of such nature, they exhibit the different type of the properties. And as you may be knowing, there are different types in the isomers, mainly the structural isomers, which is nothing but they have a different covalent arrangements
of their atoms, or the other category is cis and trans type of the isomers. They have the same covalent bond but differ in the spatial arrangement. That is the arrangement of the atom or group of atom in a particular geometry or the spatial arrangement. Or the another type, which is nothing but the enantiomers. These are actually the isomers which are mirror image of each other, and they rotate the light in a different way. Just see the right hand, this is my right hand, this is the left hand. So these are exactly mirror images. So this is like a, in the similar way, when the molecules, given molecules are mirror images of each other, they are nothing but the termed as the enantiomer, and they have the capacity to rotate the right plane polarized light either in the right direction, which is nothing but the dextro type, or the other one is the L type, that is levo rotatory type. So these are the two structures which are given here are example for the structural isomers. If you count the number of carbon atoms and hydrogen, the molecular formula is same, but their structure is different. So because of this different type of the structure, now they have the different, they are exhibiting the different physical as well as the chemical properties. Now here the cis and trans isomers. So this is nothing but because of the spatial arrangement of the atoms or group of atoms. So in the first one, you can see here the this X may be any groups there. So these groups are which are present on the same geometry or same side of the double bond, carbon-carbon double bond. Whereas in the second case, which are present on the opposite side. So the first one is cis type and the second one is the trans type. And in the enantiomer, you see here the right hand and the left hand, which are exactly mirror images of each other, just like the given structure there. So they are the mirror images of each other, but structurally they are the same, but they are in the mirror images. So because of that, again, they exhibit the different properties, particularly the rotation capacity for the plane polarized light, which will be the different. One is L isomer and another one is L for the levo and another one is for the dextrorotatory, which is nothing but the D type isomer. So I can see some questions. So should I answer the questions right now or at the later, sir? Umesh ji,
You can answer now, or you can we can take at the end of the session, sir. After.
at the end of the session. Okay, so around 5:30 after that, I'll focus
Yes,
on
sir.
answering that.
Yes, sir. Yes.
So these enantiomers. So dear students, you may ask me, what is the importance of these enantiomers? So in pharmaceutical industry, it's very, very important. Just one example I'm giving as a pharma sector, the drugs or the medicines which are available in the market, where that is nothing but the pharmaceutical chemistry or in the pharma industry, where the enantiomers are very, very important. Because any drug molecule, when any drug molecule is synthesized, they will be, one is D type and another one will be L type. And please remember, both these D and L type of molecules which are formed, they don't have the disease curing effect. So only one of the isomer will be having the biological activity or disease curing property will be only for one of the isomer. Whereas the another isomer which may not be having any biological activity, or in other words, or it may be causing some side effects or the toxic effect, or it may not be active at all. So in pharma compounds or among the pharmaceutical industry, this is first they will, after synthesizing, then they will choose the required isomer. So again, these isomers can be separated by the different type of the techniques. So these isomers are separated. Only the biologically active isomer only used for the as a drug molecule, whereas the another compound which is not at all having any activity or that may be toxic in nature, so that will be discarded. So differing the effects of enantiomer, which demonstrate that the organisms are sensitive to even the subtle variation in the molecules. You see here one such example under the pharma industry, the drug molecule ibuprofen. So mainly the ibuprofen or ibuprofen, these are the generic names, and in the market the ibuprofen is available in the name of Brufen. So I forgot the name of that company, but the commercial name is Brufen. So the ibuprofen is the actually the generic name, and this is widely used as a for pain relieving or for the relieving the or to reduce the inflammation which is caused in our body. Here the there is a R and S type of the notation, which again I am not going in detail. Again, these are purely based on the stereochemistry only. They have the different stereochemistry, or these are the two different isomers. Only the S type of the ibuprofen is having a biological activity as a pain relieving or relieving the inflammation. Whereas the R type of the enantiomer, which is not having any properties, biological activity. Similarly, the another drug, the generic name is Albuterol, which is widely used for the treating the asthma. Here, only S type of the Albuterol is, which is S type is inactive. In the previous case, S ibuprofen was, it was active. But here, S albuterol was, this particular molecule is, it is ineffective or it is not having any activity. Whereas the R type, which was inactive in the ibuprofen, but here in albuterol, the R type of the this isomer, this is actually this particular compound is effective or this is biologically active. So when there is a change in their isomer or the stereochemistry of the compound, their biological properties also are going to vary. So this is very important in the pharma industries. And at the same time, we can change the properties of organic molecules. Again, the same, that is nothing but the mainly in the drug discovery. Most of the pharmaceutical company, where they have a separate wing which is called as R&D, research and development wing, where what they do is some of the R&Ds, they are purely focusing on the developing the new drug molecules, completely new drug molecules. Whereas in some of the R&Ds, what they do is on an existing drug molecule, they will try to introduce the different type of the functional group. Thereby, they will try to enhance its biological property, and at the same time, if the drug is having any side effects or toxic effect, they will try to minimize the toxic effect. So this can be done by introducing different type of the functional groups. So a number of characteristics groups which can replace the hydrogen attached to the skeletons of the organic molecules. So if you can replace one of the hydrogen present in the skeleton, either if it is a benzene or any given heterocyclic compound, one hydrogen if you replace by I'm sorry, one hydrogen, when you replace it by the different type of the functional groups, here it is given some set of different type of the functional group like hydroxyl, which is OH group, or carbonyl, which is aldehyde group. Similarly, the carboxylic acid group or SH group, phosphate group, etc. So when we introduce those type of the group, it changes the biological property. Entirely it changes the biological as well as other properties of the drug molecule. So that is what in the pharma industries, the R&D wings, research and development wings, they are mainly working on those type of the research. These are the few of the functional groups: carboxylic acids or the acetic acid, or different type of the amines, NH2 groups, or here in the case of the glycine molecule where the NH2 group. So if you replace this by other any other functionality, then the property of the glycine also that can be changed. So in any given compound, any given organic molecule, replacing the hydrogen generally by means of other functional group that can vary its biological as well as other properties. So this is one such molecule, adenosine triphosphate. So one phosphate molecule, the adenosine triphosphate, which is the primary energy transferring molecule in the cell. So in all living organisms, including human being, the primary energy transferring molecule in the cell, that is the ATP molecule, adenosine triphosphate. So this consists of an organic molecule called adenosine. You can see here the skeleton of the adenosine. This is an organic molecule, adenosine, which is attached to a string of 3 phosphate groups. The adenosine, it has a large structure. I have just schematically presented it as an adenosine, which is bonded to 3 phosphate groups. Okay, so that is the entirely adenosine triphosphate. So it makes ATP as a single molecule. So that is a primary energy transferring molecule in any of the cell. The versatility of the carbon, carbon makes possible the great diversity of the organic molecules. The variation at the molecular level, which lies at the foundation of all the biological diversity. So these are the carbohydrates, which varieties of foodstuffs are here, bread or maize or different type of the vegetables. Huh? So in this one, mainly the carbohydrate. So this is one of the biomolecule, we can say, the carbohydrate, one of the biological or biomolecule. So if I ask how to define a carbohydrate, so different definitions. Some generally I have seen the students, they will tell it contains carbon, hydrogen and oxygen containing compound. That is not the modern definition. This was actually a century old definition, and that is wrong, because carbon, hydrogen and oxygen contain cyclic compound, which are even the different type of heterocyclic compounds. For example, furan, it's a heterocyclic compound. Carbon, hydrogen, oxygen, it's a cyclic compound, but that is not a carbohydrate. So as per the modern chemistry, the term carbohydrate is nothing but they are the polyhydroxy aldehydes or ketones. Polyhydroxy aldehydes or ketones are nothing but they are the carbohydrates. And the functions wise, the carbohydrate, they spare the proteins so that protein can concentrate on the building, repairing as well as maintaining the body tissues instead of the being used up as an energy source. And also the whatever the fat which we consume or which is formed in a body, so for the fat to be metabolized properly, carbohydrates must be present. So the presence of the carbohydrate is a very much essential for the proper metabolism of the fat molecule. If there are not enough carbohydrate, then large amount of fat are used for the energy. Carbohydrate is also necessary for the regulation of the nerve tissue and is the only source of energy for the brain. And certain type of carbohydrates, they encourage the growth of the healthy bacteria in the intestines for digestion. And some carbohydrates which are high in fiber, which helps to prevent the constipation and lowers the risk of the certain diseases such as cancer, heart disease and diabetes. Now the classification, different type of classifications are there. I am just focusing on the important, the main type of the classification only. So mainly they have been classified in the 4 different categories, and this classification is based on the when it is subjected to hydrolysis, what and how many products are going to be formed. So based on that, they have been classified into 4 categories. So first one is the monosaccharide. These are nothing but, of course, all these carbohydrates are polyhydroxyaldehydes or the polyhydroxyketones, which cannot be decomposed by the hydrolysis to give the simpler carbohydrate. And examples are glucose and fructose. So both of which have the formula C6H12O6. So these cannot be hydrolyzed further. So these are the most simplest carbohydrate, you can say. So these cannot be further hydrolyzed, and they are the simplest carbohydrate molecules. These are the monosaccharides. So glucose and the fructose as a simple examples. Then the second one, that is the disaccharide. When it is subjected to hydrolysis, if it gives 2 monosaccharides, then it is nothing but the disaccharide. So for example, the sucrose. So in our table sugar, the table sugar what we are using, it is actually a disaccharide. If you chemically hydrolyze that, it gives glucose and the fructose molecule. It gives the glucose and the fructose molecule. Similarly, the maltose, what we call the malt or the germinating cereals. So through which the malt, actually it contains the malt sugar. So when this malt sugar is subjected to hydrolysis, it gives 2 molecules of the glucose. And remember, the maltose in the free form, that means as a disaccharide, it is not having a sweetness. But when it is cleaved and when it gives the glucose molecule, it is highly sweetened. So that is the beauty of this carbohydrate. When they are bonded together, sometimes it will not give any sweetness. But when they are cleaved or when they are in the individual form, they will give the sweetness. Then the oligosaccharide, when the given carbohydrate is subjected to the hydrolysis, if it gives 2 to 9 number of the monosaccharide, then it is nothing but that is the oligosaccharide. Then the under that oligosaccharide, the trisaccharide also. That means it gives the 3 number of the monosaccharides. And the polysaccharide is one which gives more than 9 number of the monosaccharides. So best example is starch. The starch which is present in the rice which we are eating, or in the wheat, or it may be in the potato. So where the in the any plants, the food is reserved in the form of the starch. So when this starch, when we consume the rice or bread or potatoes or any starch rich food, when we consume, it undergoes the hydrolysis in our body and it produces the glucose. So that is how we'll get the energy. And in the starch is a reserve food or reserve carbohydrate in the case of the plants. And in case of the human being, in case of the human as well as other vertebrates, where the reserve food is nothing but the glycogen. So the structurally starch and the glycogen, they have the similar structure, but in the case of the animals, the reserve food is nothing but the glycogen. So during the starving period, whenever we are not getting a food, so during those period, what happens, the reserve glycogen, it slowly get liberated and it undergoes the hydrolysis and it produces the glucose. That's how we get the energy even during the our starving condition or starving period also. I'll just run the slides. Maybe I have another 6 or 7 minutes more, I think. The monosaccharide, these are the structures. You can see in the glucose molecule, the difference in glucose and fructose is Glucose is a aldehydic sugar. It has a CHO group at the terminal, you can say, whereas the fructose is a ketose sugar. So it has a C double bond O or the keto group, you can say. And these are the again D and L designation for the simple carbohydrates, which is nothing but the glyceraldehyde, which is the most simplest or the first member of the carbohydrate series. So any carbohydrate molecule, hundreds of carbohydrates, different type of the carbohydrate molecules which are known. So for you consider any carbohydrate molecule, for all those, the starting parent is, that is the glyceraldehyde. So based on the positions of the OH group in the, whether it is a glyceraldehyde or any other carbohydrate, they have been classified as D series or the L series, dextro or the L type of the isomers. These are the some other compound. In this particular table, the lower one, you can say, the lower 8 molecules are there, that is 6 carbons, lower 6 carbon compounds, which is the D-allose, D-allotrose, D-glucose. So we are, most of you are, I'm sure you are familiar with the D-glucose, but there are other compounds like D-allose, allotrose, mannose, gulose, iodose, galactose, talose. So these are all like you know, the like a brothers and sister of the glucose. Okay, so these are all are the just like brothers and sisters. So this is the D series. Then similarly L series, again 8 numbers. So that means glucose has a total 16 isomers. So how to find out that? 2 to the power n, where n is the number of this one carbon atom. Then the representation, that is through the Fisher projection. I'll just the another representation, it is called as Haworth representation, which is the cyclic structure for the this carbohydrates. Just let me skip this. This is important. In case of any of these disaccharide or trisaccharide or polysaccharide, the linkage, they are held together by every these cyclic structures. You can see they are formed by through the I think in the previous one, or in the next, here it is, the second structure, you can say the C1 and C4, they are linked through the oxygen, that is 1,4 alpha glycosidic bond. So if this oxygen is towards the, in the Haworth representation, when the oxygen on this cyclic structure, 6-membered cyclic structure, presented on the right side, right above side, and then if this glycosidic bond, if it is in the moving in the lower direction, then it is a alpha. If it is in the upper direction, then that is nothing but the beta. So this is the nothing but alpha type of the bonding. Here also, this is also nothing but the alpha type. If it is in the upper direction, then that will be the beta. So for instance, in the case of lactose, which is nothing but the milk sugar. So in the commonly in the milk, the lactose sugar is present, where it is made up of galactose and glucose molecule. So the linkage is between 1 and 4 through the beta. That means the bonding is glycosidic not in the lower direction, but it is in the upper direction. So in the first one, you can see that is the some disaccharide, that is the maltose. It is the alpha, the bonding is alpha. Whereas in the second one, you can see there is the bonding is in the upper to the lower. So that is nothing but that is the beta type. So these are the polysaccharide, and then starch is one of the best example. Starch is the one of the best example, and also the cellulose, which is commonly present in any of the plants or in the trees. So that is because of the cellulose, and the cellulose, it is an example for the polysaccharide. You may ask me one question. The animals, particularly the cow and the cattle, so they can eat any of the green leaf or the grass and it can digest, but human we can't, right? The reason is it contains the cellulose, and those cattle are blessed with one enzyme which is called as cellulase. Fortunately or unfortunately, being human beings, we don't have that cellulase enzyme. Probably if we were having that enzyme, the cellulase, then we can, just like cattle, we can graze any of the greenery which in our surrounding location, and just we can fill our stomach. Okay. So this is the glycogen structure, which is the reserve food. Umesh ji, how long I can continue?
You can continue another question.
Before
only few
going
questions,
to the
yes.
question and answer,
Yes, sir. Yes, sir.
another 5 minutes. Can I get
Yes.
5 more minutes?
Yes, sir. Please, you can.
Okay, 5 to 7 minutes. I'll try to wind up, and then we'll focus on the question and answer. So this is the glycogen. So now let us move on to the another important class of organic molecules, which are nothing but the polymers. So here the term polymers is it's made up of two words. So poly refers to the many, multiple, and mer is nothing but it's a molecule. So the term polymer, it gives the meaning that many molecules which are held together. So the term is polymer. Okay, this is about Professor Harman, who received the Nobel Prize in Chemistry during the 1953 for formulating a macromolecular structure for rubber. Rubber is it's a natural one, natural polymer. So the synthetically also which was done for the rubber, and he received the Nobel Prize. So based on the repeating units, that is he worked on this rubber molecule. So that is 2-methyl buta 1,3 butadiene. So that was the molecule. It is a monomer for the rubber molecule. Okay, 2,3-1,3 butadiene, or simply it is called as isomer. I'll just skip it. The conversion of monomers into the polymers, that process is called as polymerization. And the naturally occurring polymers and the synthetic polymers, as you know, the man-made polymers are nothing but the synthetic, whereas which are available in the nature, which are occurring in the nature, they are nothing but the natural. So we can give hundreds of examples in the synthetic. Similarly, we can list a few examples of naturally occurring polymers which are occurring in the nature, like the protein molecules. The proteins are they are a kind of a natural polymer, because the polymer, as you know, it is made up of small molecules which are held together. So in the case of proteins, these are formed by smaller molecules, are nothing but amino acids. Large number of the amino acids are chemically bonded. So proteins are natural polymers only. Similarly, the DNA molecule, that is also I'll say DNA or RNA, these are also nothing but the natural polymers only, because they have the simpler unit AGCT, adenine, guanine, cytosine, thymine, or uracil in the case of the RNA type molecules. So that is also the natural. Or the polysaccharides, the cellulose or the starch which I have seen earlier, they are also nothing but natural type of the polymers. Or in the silk, silk is present in the from the either isolated from the silkworm or the from the spider web. So this silk is basically, it is a protein. It's a protein, and these are again made up of smaller amino acid molecules. So silk or the spider web, these are also natural polymers only. So let me skip this. So various examples under the, again the natural, I'll just skip this also. Yeah, the proteins, why proteins are essential? So proteins are, these are needed for building the, these are like again building blocks for our body. So building muscles. So or the food rich in the, some of the foodstuffs which are rich in protein, either varieties of the cereals or you can see the cheese, the milk products, or the meat, or the fishes, or the eggs. So these are even including the milk, varieties of milk or the milk products. So these are all richer in the proteins, the different type of the proteins. And dear students, for your kind information, the snake venom, this is also a protein. Sir, you may be surprised to know this, right? Snake venom is also, this is also a protein, but that means there are some proteins are beneficial to us, some of the proteins which may cause harmful effect or toxic effect or poisoning effect on our body. And always remember, the venom is termed as a, in the case of snakes, there are two types. So one is venomous snake, and the term poisonous snake, it's a wrong terminology. The best scientifically correct one is the venomous snake, venomous or non-venomous. But in most of the time, commonly we call them as a poisonous snake. Oh, it's a dangerous, it's a poisonous snake. So using the term poisonous snake, it is not correct, but venomous is correct. That means there is some difference between venom and poison also. So let us see in what way the venom and the poison, how they are working. So the poison is nothing but it can cause some toxic effect just by touching or just by inhaling. But the venom is causing the toxic effect in the body when it enters in the blood. When in the venom, when it enters through the blood, then only it causes the toxic effect. If we touch the venom or if we inhale the venom, or for your kind information, suppose if there is a, if this bottle, if it is it's containing the snake venom, if I drink this, provided if I don't have any wound in my inner part of the body, so nothing will happen to me. If this is a snake venom, it's a very expensive. It is not a, the snake venom, don't consider it is a cheap product. 1 liter costs about 4 to 5 crore rupees. So the snake venom, if you drink the snake venom, provided if there is no any wounds in the body, in the inner tract of the body, then no any toxic effect. It will be, our body will just excrete it out. But at the same time, few milligrams, just 2-3 milligrams of the venom, snake venom, if it is injected in the body, then the things are different. Within few seconds, the toxic effect will be immediately, it spreads in the body. So that is the difference in venom and the poison. So amino acids, these are the basic block. And in case of amino acids, they have both amino group as well as acidic group, carboxylic group. So they are termed as amino acids. Okay. Again, the peptides and the proteins. So peptides are the smaller molecules, whereas the proteins are generally larger molecules. So the sources of the proteins already I have mentioned. So only thing is, one should have all the balanced food, means which is a balanced food in the sense which contains balanced amount of carbohydrate, balance amount of the protein, even the fat substance is also, that is also required. So don't just consume the only the fat or the only the protein, but all this balanced food, which is very much essential for all of us. And these amino acids, as I mentioned, these are the building blocks. So these are the different amino acids. About 20 amino acids are which are very much important for we all human being. But don't assume that only there are 20 amino acids. There are about more than 450 amino acids have been reported, but which are uncommon. Most common and important to the human being, about 20 amino acids. The protein denaturation. So when any of the protein, say for example the egg itself, when the egg, when you boil it, or when you are making the, while making the omelette, what happens? The which was a gelatinous type, it gets hardened, right? So that is nothing but the denaturation of the protein is taking place. So either because of the pH variation, or because of the temperature, or maybe because of the when they are exposed to the any of the chemical compounds, or when they are exposed to the electromagnetic radiation, the proteins are tend to undergo a process called denaturation. Thereby, they lose their physical as well as the chemical and the biological properties. So this is nothing but the denaturation. And remember, this denaturation, it is a non-reversible in nature. So this is about the protein digestion. Protein, yeah. So this is the last slide, and this slide was this photograph was taken in Dead Sea of Israel, Dead Sea of Israel, and for me the motivation to visit Israel is because to do my postdoctoral research, I have opted only the Israel. Later, at the later stage, I visited Singapore or Malaysia, the Canada and other countries. But first my visit was, the abroad visit was to Israel, and the motivation for me was the Dead Sea of Israel. The speciality of this one is it's not sea actually, it's a lake which is about 60 to 65 square kilometer area. It is spread across Israel, Jordan, as well as in the Palestine region. This water is such a high salinity, highly saline, about 10 times more saline than the normal sea water, and because of that, it's a highly dense. And in such a high salinity, forget about fishes and other aquatic animals, there is not a single microorganism has been reported. So because of such a high density, anybody can easily float on the water. So even if you don't know the swimming, but you can just lie down there, you'll float in the water. So when I was about 8 or 9-year-old boy, some of my one of my cousin, he was explaining all of us about there is an article in newspaper. According to that, there is a small country by name Israel. There is a Dead Sea, and anybody can float in the that sea. That was my cousin. He commented about that. And since my childhood days, it was in my mind, if I throw the stone, whether it will float or not, how it can float. So these are in my mind, and even this remained as a question till I get an opportunity to visit the Israel. I was there for about 1.5 years, so that is the motivation for me. Thank you one and all for your patiently hearing. So now it is the time for discussion. If any question and answer, questions or any clarification if you are seeking, I will try my best to answer. Over to Umesh.
Thank you very much, sir. Such a wonderful presentation. There are a few questions in the chat box, sir.
Okay.
First is one from Aishwarya BN.
Yeah, chart.
Why the molecules are called as organic molecules, and what are the three major organic molecules? I think you have covered in the
Can
your
you
lecture series also.
just opening that?
Yes, sir.
Second. What are? Can you read it?
What are three major organic molecules and why they have been called as organic molecules?
Yeah, basically the carbon-based compounds, the carbon-based which are made up of carbon skeleton, any molecule, it may be any molecule with the carbon as a skeleton, they are termed as uh, organic molecules. So not only three, we can list some hundreds of molecules. I think in my slide itself, I have given a large number of examples, starting from methane, ethane, propane, butane, etc. Slightly complicated molecules like the benzene is a one such organic molecule, but the with a different substitution on the benzene, then it comes the toluene or ethyl benzene and varieties of other functionalities. So many examples in the slides itself has been given.
Thank you, sir. There is one question I will read about it, sir, and I am not sure whether the question is correct or not.
Yeah.
The carbohydrates are necessary for the regulation of nerve tissue. Is it effective or no, sir? This is the question, sir.
Where it is, I'm not seeing that question. Carbohydrates are effective for?
Regulation of nerve tissue.
Yeah, the balanced amount of either carbohydrate and the protein, because I have mentioned in the slide, the carbohydrate along with the carbohydrate, it helps in the for the fat metabolism as well as the for the development of the for the synthesizing the protein in our body. So these are very much required in the proper balance, and the directly the carbohydrate will not affect, but the protein. So when there is a sufficient amount of protein we are not getting, then the functioning of the brain will be, it slowly it can come down. So that is what I always regularly suggest all my students, that at least during the examination, before some 10 to 15 days to your end semester examination, final examination, eat more and more as the sprouted gram it may be, or drinking the milk or the protein rich substances, so that because during the examination time the automatically your brain will be working more towards the more reading and more understanding, so a lot of energy is required for that. So that will be through the proper amount of by consuming the proteinaceous food. So to make a proper balance of the protein, the carbohydrates are also very much essential. So indirectly it is responsible.
Thank you very much, sir. Is glycosidic bond is a type of a bond?
Yes, glycoside is a linkage between the two carbohydrate molecules. It's a bond only where the oxygen atom, because what happens is OH from one side and the OH of the another molecule. There are suppose here glucose molecule, and here again they can refer the slide. Suppose here glucose molecule and here another glucose or galactose or another molecule is there, another carbohydrate. So the OH of this one at first position maybe, and the OH of this one at the fourth position. So they will react together to form the removal of the water molecule, and the first bond and first molecule and the second molecule, they will be connected through the oxygen linkage. So that is the glycosidic bond. It's a bond only.
Thank you very much, sir. There is a last question. You have mentioned about the snake venom, sir.
Yeah.
All snake venoms are protein, or what, sir? Only specific snake venoms are protein?
No, all venoms are, all the reported venoms are which are nothing but the proteins only. But poisonous compounds, it need not be. There are some rarely few of the snakes which are poisonous also. Very rare. But the king cobra, it is not a poisonous, it is a venomous. But there are some in Africa, South American Amazon forest, it is reported there are some frogs which are poisonous. That means you need not touch it even by the when you just come near to that, because of the it gives a sort of a aerial contact itself, it can be poisonous. So just by inhaling, so that is a poisonous. But in majority of the snakes which are venomous, I mean among the toxic causing snakes, in the generally majority of the snakes are non-venomous. Okay, they are not at all toxic. They are a majority. But among the venomous, the majority are venomous type. Very few are with the poisonous. Very few.
Thank you very much, sir. Now over to our CEO, Dr. M. Ramesh, sir, for few words.
Good evening, Doctor. As usual, you informative, insightful. Sure, students would have.
Sir, you are not audible, I think. Ramesh sir.
ಈ ಕೇಳಿಸ್ತಾ ಇದೆಯಾ ಡಾಕ್ಟರ್? Hello.
Yes, sir. ಕೇಳಿಸ್ತದೆ, sir.
Doctor, good evening. Thank you very much for once again a very informative and insightful talk.
Thanks.
I'm sure students will be very much benefited by your talk. This organic molecules is one of the major chapters in second PU.
Yeah.
And yeah, it will be helpful for students in CET and NEET and other competitive exams as well as their board exams.
Yes, sir. Actually, it's a very vast subject. I could not able to. I tried to cover in 50-55 minutes, but still I have to skip many of the things. It's a very vast the organic chemistry that in 11th and 12th. So I tried to give them some motivational, interesting things related to that.
Yeah, that's enough, Doctor. And we request you to give 2 questions, which will be sending in to the attendees of this
Okay, sir.
webinar, and they will be sending the answers tomorrow morning, which again will be posting it to you. You please select 2 best answers, and we'll give them cash prize.
Now I can announce it, or I can send it to the mail, sir? How?
Yeah. You can announce it now, and also please share it, because some of the students may not be able to take down immediately. And we
Or on the chat box, I can type it.
Yes, sir. Yes, sir.
I will read it and I'll chat.
Yes, sir. Yes, sir. That will be helpful.
Either keyboard or keyboard. So directly type madada. So okay, sir. The first question that is what are the mention? I'll write. Mention any 2 important. Mention any 2 important functions of carbohydrates. So this is the first question. Mention any 2 important functions of carbohydrate.
Doctor, for second question, please make it little bit complicated for them to answer, because everybody answer you would.
Okay. Can I send it through the mail, sir?
Yes, sir. Yes, sir. That will be fine, sir.
I will send it through the mail then.
The answer should be a small paragraph, sir. 3 to 5 lines. That will be fine.
Okay, then I will.
You can reframe the question.
I will remove this question and I'll give a little so that they can write it a little
Yes,
more.
sir.
I'll mail it within 5 minutes. I'll mail it.
They should have understood your talk and then written the answer.
Yeah, sure, sir. I will mail it.
Okay, Doctor. We are very grateful to you, Doctor. On behalf of KSTA, I'm immensely thankful to you for your time and wonderful talk. And again, we'll be inviting you for one more talk related to your topic of expertise in the coming days. Thank you very much.
Sure, sir. Sure. Thank you so much. Thanks for this opportunity. Soon I will mail those questions.
Okay, Doctor.
Thank you.
All participants, ಈಗ ಅರುಣ್ ಸರ್ ತುಂಬಾ ಚೆನ್ನಾಗಿ ಆರ್ಗ್ಯಾನಿಕ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಬಗ್ಗೆ ಟಾಕ್ ಕೊಟ್ಟಿದ್ದಾರೆ. ತಾವೆಲ್ಲ ಅರ್ಥ ಮಾಡ್ಕೊಂಡಿದ್ದೀರಿ ಅಂತ ಭಾವಿಸ್ತೀವಿ. ಸರ್ ಆಮೇಲೆ 2 ಕ್ವೆಶನ್ಸ್ ಮೆಸೇಜ್ ಮಾಡ್ತಾರೆ, ಅದನ್ನ ತಮ್ಗೆಲ್ಲ ಕಳಿಸ್ತೀವಿ. ತಾವು ಆನ್ಸರ್ ಮಾಡಿದ್ರೆ ಬೆಸ್ಟ್ ಆನ್ಸರ್ನ ಸರ್ರೆ ಸೆಲೆಕ್ಟ್ ಮಾಡಿ ಇಬ್ಬರಿಗೆ ಬಹುಮಾನ ಕೊಡ್ತೀವಿ. ಮತ್ತೆ ನಾಳೆ ಇನ್ನೊಂದು ಟಾಕು ಇದೇ ಸಮಯಕ್ಕಿದೆ. ತಾವೆಲ್ಲರೂ 4:15ಗೆ ಜಾಯ್ನ್ ಆಗಬೇಕು ಅಂತ ಮನವಿ. ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್.
Okay. Thank you, everyone.
ಥ್ಯಾಂಕ್ ಯು
Thank you.
ಸರ್.
Thank you, sir. Thank you very much, sir.